Get my scissors. And in here, we can actually show what we get. All that. Look at this. Infectious disease, vaccine development, infection and immunity. These are like core mission areas for UTMB. To the extent that we do biosurveillance all over the world. But we also have ambitions broader in the future of drug discovery, inside and outside of vaccines, in areas that aren't infectious disease, things like kidney disease, things like cancer.
We are really trying to understand what are the risk factors, how they talk to each other, and what are really the best druggable targets. So that's where we really are counting on automation and AI to actually really speed up the process.
I think we're entering a new era of how scientific work is done. So a really unique thing about UTMB overall, we have a BSL-4 lab here. So the fact that we're both an academic medical center but on an island allows us to basically have these dangerous samples in a good contained environment. So really in the fulfillment of that biosurveillance mission, it's not just watching what's happening, it's also trying to be one step ahead of the next pandemic. And one of the things Create Neo does is it actually collects samples from places like countries in South America, Brazil, for example.
E o que você está fazendo aqui, Pinduca?
Estou pegando o mosquito da febre amarela.
Tá bom.
Bringing some of those even to UTMB for further study, and started to build vaccine development. And alpha viruses are a family of viral entities for which there are really not good vaccine options. What are we really looking to find in these samples?
They are type 2 diabetes mice, and they're infected with a certain virus. It's extremely painful, can be fatal, and it has the potential to emerge and cause major epidemics if the conditions are right.
So UTMB is uniquely capable of covering basically the full spectrum of vaccine development, making your components of your vaccine, in which case you have to manipulate both DNA and microorganisms. These are all liquid handling manipulations that a lot of them at high scale, that you want to do consistently, you want to do reliably. You'd like to not have to do literally yourself at every single step. So it's nice to be able to systematize that. That's where the Flex is truly a workhorse in building those libraries, if you will, of the protein components of a vaccine. Can both ideate and execute for a good portion of things. Highly configurable and programmable and expressive. You can go and you can say deactivate module and you'll see that it actually goes off. So we don't have to compromise on kind of the scope of what we want to do. We'd like to push the boundary on efficient, self-improving ways to go after things like vaccine candidates, to go after things like preventive therapies.
Very few people know that approximately 10% of the total US population have some form of chronic kidney disease. The path from discovery of novel druggable targets and then ultimately first human trials is a really long, long one. So the goal of AI and our collaboration with Peter was to actually shrink that path to doesn't last 10 plus years, but maybe 2 or 3 years, and increase our success rates from maybe 10% to 20 or 30%.
Yeah, there's a tremendous role that AI plays in modern drug discovery. Things that used to be impossible to predict ahead of time can be evaluated at scale with AI.
Kidney diseases are very multifactorial. There will never be one drug, right? It will be multiple therapies for very narrow patient populations. We would like to know sooner rather than later that person is not responding to a treatment. And I think RNA-seq platform put by Peter here is going to be an essential tool to read the responsiveness to therapies.
So the thing that for us was a big attraction point to the Flex is that open source. It's not firewalled by other kinds of gatekeeping mechanisms, if you will. but importantly, it's a Python code base. So because of that, we feel like we can more freely and directly express what we want to do. And not just us, so can the AI models as well. It's kind of cool to see in physical reality what generative AI interpreted from our description of a protocol, you know? So really we're seeing like, we described to GPT what we want to do, and now it's really responsible for what's getting done. That bridges technology to a lot of users who don't primarily code. Even if you're a developer, you don't necessarily reason in code, you reason in language and you translate to code. It gives us a huge informational gain when then going and thinking about integrated systems. Because we already know what will work well and what won't work well, and we've efficiently figured that out by just having good tools to experiment in.
But that's where I think where automation comes in, that in tasks where our human characteristics might be actually leading us in a wrong way, we need AI and automation to basically take us in the right path.
What is automated science is that it is a emerging field unto itself. It is a somewhat of a paradigm shift, but it is a rich, independent, worthy field of research unto itself. We think in our own work, can we automate this thing that we're doing today? And we also think more hypothetically, what's the boundary of what can be automated? Can we bring the way we think and the way we act closer together?
I love to call them it's a magic, but I don't know how else to describe, chemists working together with enzymologists, with molecular biologists, with pathologists, with clinicians. And I think if we get together, I am very positive for the future.